I tried to take the prompt seriously for making a talk that leads to open-ended discussion. And I'm not sure that I nailed it, to be honest. So it ends up being a little bit maybe unsatisfying, or, it's a bit of a gonzo talk. So we can go ahead and get into it. I don't know the format here. I'm happy if people want to interrupt. It's 20 minutes long. But do whatever you want to do. Ask questions if you want to ask. I have no particular need to get to the end of my slides. So everyone can...
Feel free to stop. Feel free to ask questions.
Let's do
or throw stuff at me, whatever you need to do. Okay. Okay, great. So the title of this session is whatever it is, quantitative frameworks for state fate prediction. So, those words can be defined in various ways. The particular form of state and fate that I'm going to be talking about live in genomics land, in particular, where we're talking about state as determined from a gene expression profile. So we'll call X a vector of gene expression levels, and we're looking at the time derivative of X. And in the single cell RNA sequencing field, many of you may be aware this is called sort of RNA velocity analysis, where we observe these sorts of vectors across an entire cloud of points, and somehow we can stitch those together to represent fate. So for those who aren't familiar, or those who just want a refresher, also to start drawing some pictures that I'll refer back to, the basic idea of RNA velocity analysis is it's a derivative of single-cell RNA sequencing. So in single-cell RNA sequencing, of course, we end up with snapshots of gene expression profiles from a bunch of individual points. So all the dots in the bottom left plot there represent single cells and their gene expression profiles. And the idea of RNA velocity is to layer on top of that some sort of rich dynamical information. So now, instead of just a snapshot of the current state of each cell, we also have a vector that's tell us how that state is changing. So for each gene expression level, we've got a rate of change. And I'm not really going to talk about the ways that we estimate these velocity vectors. You can see from the picture there, one of the ways that we do it is by labeling individual transcripts during a particular window of time, and we can do things by measuring inside the window or outside the window and looking at decay rates and so on. There's a couple different classes of methods to determine RNA velocity vectors. This, in my opinion, is one of the more quantitatively viable versions. But again, the idea is that we can use these velocity vectors to sort of project forward to the future state of each individual cell. And then there's another layer of analysis that can go on top of this that people refer to as vector field mapping, where you take an entire cloud of points, and for each point now you have paired data. You've got a gene expression profile and you've got a velocity vector, and the idea is to learn some sort of function that allows you to fill in all these pretty looking arrows everywhere in the field where you haven't actually observed data. Okay, and then from that, the idea is you can somehow do lots of interesting analysis. You can find stable points, attractors, saddles, whatever you like, and in general do sort of hope to obtain a better understanding of the regulators of cell fate, and eventually maybe a quantitative and causal insight into cell fate decisions and trajectories. Okay, so that's most of what I'm going to be talking about is living in RNA velocity land. And these methods have really been very widely adapted and adopted. So since 2021, I found about 39 major methods papers, which form sort of the methodological foundation for the field. And those papers have been cited in about 5,000 downstream applications in that relatively short period of time. As I said, probably a couple more published in the last 5 minutes since this talk started. All right, so the title of this talk is weird. The first part says against prediction. I don't really know what I mean by that, but I'm supposed to stir up discussion so you all can have your own thoughts. So we're supposed to be talking here a little bit about how this is a predictive framework. So I mentioned before this vector field mapping task. And the idea of the vector field mapping is to learn this function f, where we have input data x that tells us the current state of the cell, and we're learning a function that outputs a velocity vector. Okay, and that allows you to predict fate from state. So from the beginning, this entire operation is formulated essentially as a prediction problem. You give me an unseen state, and I can tell you its fate. okay? Because if it's just the points we've already observed, I don't need a function. I already have that data in hand. So, I say here the bottom bullet point, I'm going to get to in a second, two sort of quibbles with this. But the first big quibble is that I'm not so sure the functions we learn are actually ever correct. So we're going to return to that at the end, where I'm going to show you a demonstration of how you get incredibly beautiful, just absolutely aesthetically pleasing plots that are entirely artifactual from some very basic things that have been left out of the formulation. So we're going to come back to the end. So the major quibble, I guess, is that they can very easily have systematic errors. Yeah.
Just when you say predicting fate,
Yeah.
you really are meaning, I'm just starting it, right?
Yeah, get into
A
it.
good example. Okay,
Yeah.
so you really mean sort of the larger process of what you learn from, really from the derivative, you just learn one tiny time step.
Yeah, yeah. And then you stitch all those together.
Talking about how that gets stitched together
Yeah.
as a way of predicting fate.
Yeah, yeah. Well, again, the sort of velocity vector tells you the immediate future of the cell. So that's already fate in some sense. And then you can also stitch together across all the points that are the immediate futures of the subsequent points to get a sort of longer term trajectory. Yeah. So yeah, there's a prediction that's an immediate future, and then you can stitch all those together to get predictions over longer term trajectories, supposedly. Yeah, please.
So I'm curious, like how you and others think about like mechanisms by which like a cell stores RNA, like the most recent RNA velocity kind of things use these like metabolic labeling strategies that
Yeah.
I just alluded to, right?
Yeah.
But what happens when you have a cell state that is buffered in some way, right? Where like say RNA is sitting in a granule such that you poison transcription and now it's going to start exporting the spliced RNAs from that granule. Like how do you and other single cell folks think about these kinds of weird edge cases which seem to be more and more common,
right?
And maybe we thought.
Okay, so it's a great question, and my immediate answer is literally what happens is that first sub-bullet. We learn F by arbitrary ML method applied to mountain of data. And so how is that accounted for is not at all. Okay, is the answer. And it would be lovely to do so. So I'm going to show some examples that are not that aspect of biology, but that are other aspects of biology. And so again, the first part of the now, it's not to say that you couldn't or people don't do specific things, but the very general framework is this predictive framework, and the predictions are sort of agnostic to mechanism, which is like the entire world right now. It's like agnostic to truth, okay? Postmodern out there. Scary time to live. Okay, so we're going to come back to that in just a second. Okay, so now my first quibble with this is sort of very minor point. I don't care about this so much because in just one slide, I'm going to tell you how I don't want to do this at all. But if you're going to do this anyway, be in the business, and we thought about doing this, okay? I spent a lot of time thinking about this. Be in the business of learning these functions F, and it would be cool if we owned the true function, if we knew the true vector field. That seems like it'd be really cool. Now, it's a little bit different from other areas of data science in biology, just applied to observational data, because here, if you own sort of the true vector field function, it actually feels a lot richer than other times that we have statistical descriptions of our data, because this one is sort of intimately tied to the causal structure of the system. The true vector field map should describe how the current state of every gene is affecting the future state of every other gene in the system. So the causal structure should be intimately built in there. But the way that the functions are learned right now, there's no reason to really expect them to be able to capture that structure. It's arbitrary ML models applied to observational data. And so that just doesn't sound like a good recipe to be able to recapitulate the complex regulatory structure across multiple different states. So that's my first quibble, is that the way that this is set up, the sort of causal structure should be baked in there, but the way that we run the experiments and design them, I don't really have any expectation that they will actually be able to recapitulate that.
Is there a bigger quibble that X may not be a sufficient set of variables
Totally.
or cells that determine the RNA velocity?
Totally.
Okay, that seems like it's a bigger quibble than the ones you're mentioning.
Multiple quibbles
Yeah.
we've had here. Maybe quibble's not even the right word. We could quibble about quibble. Okay, let's keep going. So a second quibble, okay, and that's even sort of a broader one that gets back to the title of the talk, is I'm not quite sure what we're getting out of this. So again, one of the hopes of this, I'm not so sure that we really care about making predictions. Ideally, we're learning something about the nature of developmental systems or other sort of dynamic biological processes here. And I'm part of this is to gin up discussion. I'm not trying to disparage any work. We've done all of this stuff that I am, sort of saying. I'm not quite so sure this is well founded. Okay, so I'm trying to gin up discussion here. So supposedly by learning this function f, we're going to be able to learn something about what regulates fate. And in particular, that takes the form of looking at the Jacobian of this function here. It tells you in every single state that we could imagine, at least that we've fit somewhere in the neighborhood of the cloud of points that we've observed, which genes are causing changes in the future state of other genes. Okay, and that's the form of sort of understanding of regulation that immediately pops out of this framework, is that we end up with a big lookup table over the Jacobian, right? Because it's different everywhere in space. It says if you're in this gene expression state, gene A is causing gene B to go up. And if you're in a different gene expression state, the regulatory relations are a little bit different. And you end up with a big lookup over the Jacobian everywhere in space. Yeah.
So forgetting the observation, from my angle into this, usually this is, so you're thinking about methods where they actually look at the joint distribution in some way of different genes, or the interactions have some, much of this is just done on a gene by gene basis, right? And so the only thing that affects the next state of mRNA of a gene is the previous state of that gene. So you could try to learn something about how the genes affect each other, but it's not baked into that.
No, it is.
Differential equation. It's baked into the biology, but it's not baked into usually not baked into the mechanism.
Not explicit, but it's implicitly in there. That's what this function tells you. It tells you how it's a function over all the genes. And it tells you how every gene is affecting the future state of every other gene. It's implicitly in there. Sorry. Yeah.
So
it has to be.
This guy, we had a hand up back here first.
I'm confused now.
Yeah.
Are you talking about transient or a snapshot of a state in an attractor state? Because then you don't have any information anyway. And a lot of the RNA-seq are in it's an equilibrium in attractor state, right?
Yeah, or they're near some very canalized trajectory. Yeah, and so that's again part of the point from the previous slide is you actually don't, from just observational data, the most informative points you never get because you're either just in an attractor state or you're in a canal along a very canalized trajectory. And the most informative stuff is actually off of that somewhere, which is sort of why you need to incorporate perturbational data. Yeah, please.
I can't help to address because it seems that you are talking about my work. Yes, what you said,
okay,
yes,
yeah, sorry.
Analyze, there's no problem, right? There's some region you cannot, right? You don't have data, you cannot really help.
Yeah.
That's no problem. And also here, talk about the Jacobian.
Yeah.
Who said that Jacobian, you only use Jacobian to infer the dynamics?
Sorry?
Who said that?
Who said that?
Yeah. The Jacobian, you said you look at the Jacobian to find the relation things. This is a lot of misunderstanding in the literature.
This is not, sorry, well, I don't understand. What's the problem? You learn a function and I can look at the
But the Jacobian alone cannot really tell you.
Oh, true. Yeah, that's fine.
Yeah.
The Jacobian alone cannot really tell you the full. Yeah, that's fine.
And that's what I don't know that people talk about this. They're like, okay, you just look at the Jacobian, you will tell you the things.
That's true. But I guess in general, my sort of problem is that, or the discussion topic I want to raise, we've done this stuff too. Okay. The discussion topic I want to raise is that this is formulated from the outset as much more of a predictive task than some alternative formulations. And the issue that I want to raise for discussion is that formulation as a predictive task like this, for me, as the bottom polemic states, often does not seem like the direct path to understanding the nature of things that alternative formulations might give us. Okay, just a point for discussion. It comes across as trying to disparage, and I screwed up my gonzo talk a little bit. So just to give an alternative formulation, and here I'm not, you can read the disclaimer at the bottom of the slide, but just again to propose something that's just to show you what I mean by something that's not formulated as a predictive problem. Suppose that instead we think of fate as being specified from a product of some bias over the field and a series of constraints. Okay, so think of the bias as being like a direction that points sort of downhill. Okay, and the constraints are like bumper rails. Okay, you sort of can't move past the constraints. To me, this seems like understanding the nature. So this is like has nothing to do with prediction. We don't understand at every single point in space which gene is doing something to every other gene, and that changes everywhere we go. There's a relatively small number of things that we need to understand, and it seems like interrogating the nature of this bias term and the nature of the constraints might be very fruitful for understanding the underlying biology of the system. Okay, so again, I just bring this up not because I'm trying to say people have or have not done it or that I think things actually work this way, but just to illustrate an alternative to formulating as a prediction problem. That's point number one. And point number two, which is for the remainder of talk, to introduce the idea of constraints, which are one of the major things that's missing from the framework that I've been describing so far. We learned essentially an unconstrained, it's constrained by the data, I suppose, but we're learning an essentially unconstrained function f that maps from a gene expression state to a velocity vector. So the rest of the talk is going to be trying to think a little bit more about constraints in this context that I've introduced so far. So first up in that, we started thinking about this a couple years ago, and I'm going to sort of recapitulate my wandering path through this idea of constraints in RNA velocity analysis. And there's going to be a tiny sprinkling of results in there a little bit. I already told you it's going to be an unsatisfying talk, so hopefully nobody's let down too much by the end of it. So the first thing to think about is, is there a constraint that says how fast can a cell move? If I sum up the velocity value of every single gene, is there a physiological constraint to that? Can a cell move arbitrarily fast in transcriptional space? If not, what sets the limit? Okay, so that's sort of first question. Like, can those arrows, if I take an arrow, can it have, it's not exactly this, but an arbitrary magnitude? Okay, so that's a type of thing that's sort of not baked into what I set up so far as the initial problem. All right, and you could approach this through different ways. Here's how I started thinking about it. One potential limit to the total, I'm going to call the thing I've been mentioning so far the total velocity of a cell. One potential limit derives from flux balance through the nucleotide pool. So there are a couple different demands on the NTP pool in a cell, and I should say that the fluxes in all these different demands are really massive compared to the size of the pool. So we can tell immediately from that that the fluxes have to balance quite closely. So the nucleotide pool has a couple things that consume it. One of the sources of consumption is we eat up NTPs when we synthesize RNA. That RNA can be degraded, and those can then be salvaged to be returned back into the NTP pool. All that stuff is shown in the purple box on the left. You can also de novo synthesize NTPs. That's another source of NTPs. NTPs are also consumed via conversion into DNTPs.
But this is a good system but the dangers of what you're doing is the mechanism changes a lot depending on the pool. So if there's enough nucleotide, then things just keep producing no things. If you run out of it, you have to go through a complete different cellular mechanism that do synthesis of it.
Yeah.
If you put all in a single constraint, you may run into trouble.
Yeah. So again, part of what I want to do here is sort of introduce my wandering path to the actual thing that I did. So here, this is figure 5 from a grant proposal or something, not from any sort of actual results where I did something useful that contributed to humanity. Okay, so again, we can consume via a couple different routes, and we can think that these generally should balance, or at least that's one way to start pulling a thread on some interesting stuff that might lead us to a constraint. 3 minutes. Okay, I have 47 more slides. Okay, so if you write down the balance between those terms, that's this first line here. We can produce via de novo synthesis. We can salvage, which has to do with degradation of RNA times the abundance of RNA. And that should balance, those are both inputs. The inputs should balance the rate at which we consume them. If you rearrange those terms, you get on one side, the total velocity of the cell should equal these sorts of non-RNA related fluxes. Okay, so that's interesting. So I started thinking about this I can measure because I can get a crappy estimate of the RNA velocity in every cell, so I can sort of measure it. These maybe are a little bit less obvious, but at least one of them is related to the growth in a cell. So that's one thing I started to think about is as the cell grows more quickly, it's going to have to consume more of the NTP pool to double the DNA content of the cell. So I started thinking as that growth rate goes up, what happens to the constraint on the velocity of mRNA in the cell? And that led us down a different path to say, okay, well, let's think about in general, and this is actually ancient biology, but somehow has been left out of the RNA velocity framework. There's a homeostatic constraint on the RNA velocity in the cell. When cells grow, they double in volume and they double their RNA content. So a gene that starts out with 2 transcripts by the end of the cell cycle should have 4 transcripts. A gene that starts out with 6 transcripts by the end of the cell cycle should have 12 transcripts. So that means the sort of homeostatic velocity is necessarily positive. And it's positive in a way that introduces a relationship between the abundance of a gene and the average velocity of a gene over the cell cycle. Genes at higher abundance need a higher velocity because they have more transcripts to double over the cell cycle than a gene with a low abundance. And this slope of the line gives you the growth rate of the cell. Okay, so that's sort of our homeostatic expectation.
Can I ask you, want to clarify? You say level. What do you mean level? Count?
Counts.
Or count?
Counts.
Not concentrations.
Concentrations we don't measure. We can come back to this point. A lot of TPMs are not
When you talk
concentrations.
about that, right? You see that, right? You see, because you talk about the rate, we're talking about concentrations.
Well, we can measure sort of count. We don't measure concentrations. We could talk about this in a second, but people often put things in units of TPMs for the single cell aficionados in the crowd. Not a good proxy for concentrations. I hear a timer going off. 30 seconds. Sorry, we'll come back to the thing at the end. So it's just to say that growth mostly hasn't been written down in this framework, and it introduces a couple different artifacts. The first thing that happens is what's called the degradation term gets misinterpreted. This has been sort of introduced in many other fields of biology over time, but by far not the first time that people have noticed this. But in RNA velocity, it seems to not be appreciated. That the thing we call the degradation rate is actually an unknown mixture of the growth rate of a cell and the actual degradation rate of RNA. But perhaps the most important thing to show is if you don't account for the fact that the homeostatic expectation is not a velocity of 0, but actually needs to account for the doubling of the cell over time, these vector field plots that you end up with can be sort of systematically wacky. Okay, and this happens even if you measure stuff in TPM units. Okay, it happens there too. We have lots of data on this. But here I'm showing you the results of a simulation. We just simulated cells that aren't doing anything except for doubling over the cell cycle. And then we ran standard RNA velocity packages to generate one of these plots that shows stuff. So the only thing that's happening in the ground truth is that cells are proceeding through the cell cycle. So they're kind of going from right to left or whatever direction my arrows go there.
But that's another problem, right?
Yeah, the legend's mixing colors. That's
light green more or less big?
This is beginning of the cell cycle. This is end of the cell cycle. Okay, so that's the ground truth. And then if you sort of run the standard RNA velocity on sort of simulated observational data here, you get these beautiful plots, absolutely beautiful plots that have nothing to do with what the ground truth does. It seems to imply that there's some sort of ridge decision boundary, but it introduces absolutely artifactual trends. We've got lots of other simulations. There's a preprint that's up that has all this stuff. And we've shown that this happens in every publicly available data set that we can get our hands on as well. Okay, I think I passed my 30 seconds. So the remaining 42 slides will have to wait. But thank you so much. Yeah.
But I think